Good afternoon, everyone, and thank you so much for being here. It's my absolute honor to be presenting our work. Today, I'm going to be discussing one of the biggest challenges in generating clinical evidence and discussing how my team is leveraging innovations in agentic AI paired with deep clinical expertise to accelerate clinical innovation. This is the major challenge that we face in generating clinical evidence. Despite the fact that in the United States, all healthcare institutions are now on electronic medical records, with the goal of these requirements being that we could unlock all this information for big data and evidence generation. In many ways, we still document the same way we have for hundreds of years. Most of the most important information that we document on patients is often present only in unstructured clinical notes, radiology reports, or pathology reports, and not accessible for automated scalable analysis. This is further complicated by the fact that when information is documented this way, it's not reliably standardized, and we can't link across data sets. This is a major barrier for clinical trial conduct and real-world evidence generation, for example, for pharmacovigilance. And this is one of the consequences for clinical trials. Conducting clinical trials is very expensive, time-intensive, and because of the challenges of handling clinical data, sometimes compromises quality. Poor accrual to clinical trials is one of the major reasons trials fail. For example, in oncology, over 20% of trials terminate because they can't accrue enough patients. And we believe that one of the major driving factors and ways to address this challenge in particular is to make it easier for sites and sponsors to collect data from electronic health records. If we could lower the barrier to extracting and standardizing information from clinical documentation, we could make it faster and more efficient for sites to get the information needed out of the data to report on trials, broaden the participation base, and make innovations more scalable and accessible throughout the country. Here's an example of a typical workflow for clinical trials, but frankly, for most real-world evidence tasks, the workflow is quite similar. Research coordinators or research nurses at a trial site scours through the raw data on a trial participant and extracts the relevant information needed to report on a trial. That's then verified by a physician or APP, and study monitors then come in and audit. Finally, it's approved. Each step of this process takes a long time, and then there are backups between each stage. This means that trial monitors cannot find deviations until after the fact, so there's no way to proactively intervene when you might have known that, for example, you hit a dose-limiting toxicity too early. In addition, these manual processes limit the reuse of data for long-term pharmacovigilance and ongoing real-world evidence generation. There are existing solutions for EHR to EDC extraction, but importantly, none handle unstructured data and do not provide direct source document verification because of this. Because so much of the information for clinical trial reporting is, in some cases, only documented in unstructured clinical notes, existing solutions do not meet the needs for sponsors and sites. Our solution is to develop agentic AI systems to automate reporting, including source document verification. These systems are not meant to replace humans. They are meant to augment humans participating in trials or other real-world evidence generation activities. They automate the process of surfacing information from the electronic health records, so then humans can then verify and move that into the EDC. For healthcare institutions, this increases their capacity to participate in trials and leverage existing data for real-world evidence. For CROs, this can streamline operations, auditing, and secondary use of existing data sets. And for sponsors, this can reduce costs, enhance data quality, and improve trial success rates. We've started with extracting immunotherapy-related adverse events. We chose to start with adverse events because these are one of the most time-intensive and nuanced pieces of information to extract from electronic health records. And almost all trials need to report on adverse events. We've built on over 10 years of experience in natural language processing focused on information extraction from medical records, paired with deep expertise from clinical trial experts at MGB on trial reporting. We've developed agentic systems of LLMs which collaborate together and use tools such as drawing from guidelines and knowledge bases to extract the adverse event, conduct the severity grading, and attribution. Our agentic system has been developed and prospectively validated on a uniquely large gold-labeled data set from Mass General Brigham. And our results are really promising. When research coordinators use our system, their accuracy at extracting adverse events increases. This is accuracy compared to an expert oncologist. In fact, when they use our system, their accuracy approaches the performance of the expert oncologist who created our gold-labeled data set. At the same time, they're more efficient. When research coordinators use our system, it takes them half the time to extract information from a note compared to when they do it manually. Now, we know that for clinical trials, data quality cannot be compromised. And so because of this, we've designed our system with human verification and regulatory compliance in mind. Now, this is kind of a simplified example of the current manual standard where research coordinators, nurses read raw medical data and determine if an outcome is present, for example, an adverse event. Our system keeps that human oversight in place, but enhances it with automation. You might ask, why can't we just use a simple LLM call to do this? There's two reasons. One, we found that even leading LLMs cannot conduct the type of standardization and reasoning needed for these clinically nuanced endpoints. And also, simple determinations do not provide any support to the extractors in reviewing and auditing. To solve for this, we've worked hand in hand with research coordinators and clinical trialists to design an interface that supports oversight and compliance. Our system not only extracts the adverse event information, but also highlights the relevant information from the data to support that evidence and provides an explanation. Ultimately, our vision is to release the trial reporting bottleneck so that breakthroughs can reach patients faster. And we believe that these technologies could support sites, CROs, and sponsors. So far, we've developed a proof of concept in immune-related adverse events, and it is now being run prospectively at Mass General Brigham to support a biobanking trial. We now seek partners to de-risk our efforts and build it out for commercial use. And there are two avenues that we envision. One is funding to build out specifically the pharmacovigilance and adverse event pipeline. There also is a larger opportunity to extend this to broader agentic automation of trial operations in the form of a new company. Thank you so much for your time.